Hello friends, welcome back to a new video, that is MCQs on biotechnology, and this is the part 9 of this series of videos on biotechnology. Okay, so the first question is question number 161 of this video. So the first one is the enzyme primarily used by prokaryotes for synthesis of DNA is or are called as option A restriction endonuclease, option B DNA polymerase, option C phosphodiesterase, or option D any of the above. So the right answer is option B, that is DNA polymerase. So the enzyme primarily used by prokaryotes for synthesis of DNA is or are called as DNA polymerase. Okay, so let's move on to next one, that is 162. So those enzymes that are structurally different but catalytically alike are option A duplicate enzymes, option B isozymes, option B abzymes, option D ribozymes. So the right answer is option B, that is isozymes. So those enzymes that are structurally different but catalytically alike are known as isozymes. So let's move on to question number 163. That is, a novel type 2 restriction enzyme has been isolated from a thermophilic bacteria. Okay, so this restriction endonuclease recognizes a 5' ATAA triple N TT 3N. This sequence they can understand or they can recognize. So here N, this 3N means any nucleotide. So here N means any nucleotide, and they cut after third A, that is this. They can cut after this. After this means here they can cut. So they can cut after third A in the above sequence. So what is the fate of DNA after restriction digestion? So the options are option A, a 3 nucleotide long 5' overhang, or option B, a 7 nucleotide long 5' overhang, or option C, a 4 nucleotide long 5' overhang, or a 3 nucleotide long 3' overhang. So the right answer about the DNA after restriction digestion of this sequence, that is option A, that is a 3 nucleotide long 5' overhang we will get. So here the 5' and the cut is here, that is ATA. This 5' overhang we will get after restriction endonuclease digestion. Okay, so let's move on to question number 164. That is, polynucleotide ligase joins two DNA molecules together by forming a covalent bond between option A 2 OH groups, option B 2 phosphate groups, option C complementary nucleotides on opposite strands, or option D a 3' OH group and a 5' phosphate group. So the right answer about the polynucleotide ligase joins two DNA molecules together by forming a covalent bond between option D, that is a 3' OH group and a 5' phosphate group. So ligase can combine both of these groups, that is 3' OH group and 5' phosphate group of DNA molecule. Okay, so let's move on to question number 165. That is, RNA can be used as template when option A, it is artificial, option B, converted to cDNA, or option C, it is impossible, or option D, cannot be predicted. So the RNA can be used as template when, and the right answer is option B, that is converted to cDNA. So RNA can be used as template when it is converted to cDNA or complementary DNA. Okay, so option B is the right one. So let's move on to 166. The Vibrio fishery has ability to convert chemical energy to radiant energy during process of bioluminescence. Justify your answer. So the options are option A, first law of thermodynamics, option B, second law of thermodynamics, option C third law of thermodynamics, or option D all of the above. So the right one is option A, that is first law of thermodynamics. So the Vibrio fishery has ability to convert chemical energy to radiant energy during process of bioluminescence by the law, that is the first law of thermodynamics. Okay. So let's move on to question number 167. So how many molecules of CO2 is or are given off in 10 turns of Krebs cycle? After 10 turns of Krebs cycle, how many CO2 we will get? And the options are 40, option B 20, option C 80, option D 10. And the right one is option B, that is 20. So after 20 CO2 molecules we will get after 10 turns of Krebs cycle. Okay, so next question देखेंगे. Next is the glycolysis enzymes are located in option A nucleolus, option B vacuole, option B plasmodesmata, option D cytoplasm, and the right one is option D, that is cytoplasm. So the glycolysis enzymes or the enzymes which are involved in glycolysis process are located in cytoplasm. Okay, so next one is how much is oxygen requirement of for fermenting 1 glucose to ethanol and CO2? And the options are option A nil or 0, or option B at least 4, option C at least 1, option D 2 only. So the right one is option A, that is 0 or nil. So no oxygen requirement of for fermenting 1 glucose to ethanol and CO2. Okay, so let's move on to 170. That is, which of the following can be applications of protoplast? And the options are option A hybrid cell formation, option B modification of genetic information, option C study of industrial application, option D all of the above. So the right one is option D, that is all of the above. So all of the above, these are the applications of protoplast. Okay, so next one is 171. So which of the following is or are continuous cell line? So options are option A KB cell line, or option B HEP2, option C HeLa, or option D all of the above can be used as continuous cell line. So the right answer is option D, that is all of the above are right. So this KB cell line, HEP2 and HeLa cells can be used as continuous cell line. Okay, so let's move on to 172. That is the SD sequence is or are option A start codons, option B nonsense sequence, option C sequence known for binding ribosome, option D none of the above, and the right answer is option D, that is none of the above. So the next one is 173. That is, which of the following is or are nonsense codon? And options are UAA and UAG, or option B UGA, or option C both A and B, or option D neither A nor B. So the right answer is option C, that is both A and B. So UAA, UAG and UGA, these all three are nonsense codon, okay, or stop codon. So the next question is 174. That is the phenomenon of losing or deleting plasmid from cell is or are referred as option A deplasmidization, or option B curing, or option C plasmidolysis, or option D, that is plasmid burst. So the right answer is option B, that is curing. So the phenomenon of losing or deleting plasmid from cell is or are referred as deplasmidization, or sorry, the phenomenon of losing or deleting plasmid from cell is or are referred as curing. Okay, so the next question is 175. That is, which of the following differentiate between microarray and gene fusion? And options are: microarray uses DNA from different gene. Option B, microarray is not constructed by gene cloning. Or option C, microarray gives direct measurement of mRNA level. Or option D, all of the above. And the right one is option D, that is all of the above. So all of these are correct about the differentiate between microarray and gene fusion. Okay, so let's move on to next question, that is 176. So which of the following is or are responsible for multiple drug resistance, mainly in tuberculosis organisms? And the options are: transformation, option B conjugation, option C mutation, or option D iron deficiency and fudge infection. So the right one is option C, that is mutation. So mutation is or are responsible for multiple drug resistance, mainly in tuberculosis organisms. So the next one is 177. So the bacterial cell capable of taking outside naked DNA is referred as: option A complementary, option B complement, option C cured, option D saprophyte. And the right answer is option B, that is complement. So the bacterial cell capable of taking outside naked DNA is referred as complement. Okay, so next one is 178. It is, which plasmid plays vital role in conjugation process? And the options are: option A plasmid C, option B plasmid F, option B plasmid F, option C plasmid R, or option D all of the above equally. So the right answer is option B, that is plasmid F. So plasmid F plays vital role in conjugation process. F for fertility. Okay, so move on to next one, that is 179. So the location of genes in the genome can be studied easily by: option A transformation, option B electrophoresis, option C annotation, or option D genomics. And the right one is option C, that is annotation. So the location of genes in the genome can be studied easily by annotation. Okay, so let's move on to the last question of this video, that is, in which of the following phenomenon both DNA strand serve as template? And options are: option A photoreactivation, option B replication, option C excision repair, option D all of the above. And the right one is option B, that is replication. So by in replication process both DNA strands serve as template. Okay, so these are all 20 questions of this part 8 video on biotechnology. Okay, so thank you for watching this video.